Hi, my name is Dr. Scott Coffin. I'm a research scientist with the California State Water Resources Control Board, and today I'll be talking to you about managing microplastics, lessons learned from California's research and regulations. The plastic waste crisis has recently gained a lot of attention in the media. Everywhere we look, we seem to find plastic, from the deepest part of the ocean to the tallest mountain in the world. Plastic degrades slowly in the environment, breaking into smaller and smaller particles along the way. Plastic particles can make their way into water through multiple routes, including through stormwater runoff, wastewater treatment plant emissions, atmospheric deposition, and littering. Plastic particles may become buried and resuspended in the water column, and in some cases, plastic can make its way into drinking water supplies through surface waters. To address public concerns of microplastics, the California legislature passed two groundbreaking bills in 2018. Senate Bill 1263 is aimed at aquatic ecosystems and requires the Ocean Protection Council and State Water Board to initiate a statewide microplastic strategy by 2022, and by 2026 develop a risk assessment framework, standardize analytical methods, establish baseline occurrence data in the environment, investigate sources and pathways, and recommend source reduction strategies back to the legislature. The first study to monitor for microplastics in tap water found these particles in 83% of samples worldwide and in 94% of samples in the United States. These findings prompted California to pass Senate Bill 1422, which requires the State Water Board to define microplastics by July 1st of 2020, and by July 1st of 2021 requires that we adopt a standardized method for monitoring, a plan for 4 years of testing and analysis, consider adopting a health-based guidance level to aid consumers in interpreting concentrations in their drinking water, and accredit laboratories so that we can feel reliable about the results. Defining microplastics is no trivial task, as this contaminant suite includes billions of different combinations of particles of different chemical compositions, shapes, sizes, colors, and associated contaminants. Despite these significant challenges and a lack of a harmonized definition, the State Water Board adopted the world's first regulatory definition of microplastics in 2020. This definition is broad and includes particles between 1 nanometer and less than 5,000 micrometers. Armed with a legal definition of microplastics, we can now investigate the risks to humans and aquatic ecosystems. To meet the legislative mandates to assess risks, we convened a health effects workshop with global experts. We performed a meta-analysis of the available toxicity literature and examined relationships between particle characteristics and toxicity, adverse effect pathways, and dose response, and attempted to derive thresholds for aquatic ecosystems and human health. Microplastics are a complex mixture of particles and chemicals, and can present risks to biota through both of these traits. While environmental microplastics are diverse in shape, size, and polymer type, laboratory effects studies are typically only conducted on a single particle type, or a distribution of types that may not be representative of those that are in the environment. To reliably compare between laboratory effect data and environmental monitoring data, we first need to understand the particle trait responsible for the toxicity. This is called an ecologically relevant metric, and for aquatic organisms, we know that food dilution is responsible for toxicity, which can be explained by the volume of the particle. We must restrict the bioavailability by those particles that can actually be ingested by each organism. We also know that tissue translocation-mediated effects like oxidative stress and inflammation can cause toxicity, and this is predicted by the surface area of the particles. Of course, restricted in bioavailability by the particles that are actually capable of translocating. Once we understand the ecologically relevant metric, we can then compare between the particles used in the lab and those in the environment using first principles, environmental data, and probability distributions. Now I'll describe how we applied this framework to assess risks to aquatic ecosystems. Starting with a literature search, we found 167 in vivo aquatic microplastics toxicity studies with 117 species. We then developed screening criteria to assess the reliability of these studies, filtering out studies that were not reliable or relevant for risk assessment. And we're left with 22 studies with toxicity information for 16 different species. We then subjected these studies to additional scrutiny through expert review, aligned the data using ecologically relevant metrics and probability distributions, and then derived species sensitivity distributions. These aligned effect thresholds can then be used to predict risks to an entire ecosystem using a species sensitivity distribution approach. We chose to use the 5th and 10th percentile of modeled species affected to set our risk-based thresholds. Our working group developed a 4-tier management framework, with each tier corresponding to higher concern that effects are occurring and of higher severity, and each corresponding to higher levels of management. For instance, exceedances of the 1st tier, we would recommend additional investigative monitoring, whereas exceedances of the 4th tier, we would recommend more stringent measures, such as source control measures. We developed quantitative aquatic ecosystem thresholds for each of these tiers for 2 ecologically relevant metrics, including food dilution and tissue translocation. The concentrations shown here are based on a particle size distribution between 1 and 5,000 micrometers, allowing reliable comparison to environmental monitoring data within that size range. We asked the experts to rate their confidence in the framework and the analytical process used to derive these thresholds. Overall, they expressed high confidence in both approaches. When asked about the thresholds themselves, the experts expressed low to medium confidence, citing uncertainties in the underlying studies. Recognizing that other jurisdictions may prefer to take different approaches to deriving thresholds using species sensitivity distributions, we developed an open source web-based application that allows users to rapidly derive custom ecological risk thresholds tailored to their management needs and specific ecosystems they're trying to protect. For example, one may only want to include freshwater species in their assessment, or perhaps just commercially relevant species. This tool allows users to align the particle-based toxicity data to ecologically relevant metrics based on site-specific monitoring. For instance, some regions may have a higher proportion of fibers or tire wear particles than others, and the assessor can integrate this data into their site-specific risk assessment. This is just one of many tools available in our app. Other tools include data visualizations, Monte Carlo particle simulators, a machine learning model that predicts toxicities of microplastics, and the ability for users to upload published toxicity data. We applied this risk assessment framework to characterize risks of microplastics in San Francisco Bay, California. Surface water microplastics were collected using a mantitol net by the San Francisco Estuary Institute and 5 gyres. A number of data cleanup and corrections needed to occur to reliably compare between environmental monitoring data and effect thresholds. Sampling and analysis techniques included systematic biases that could be accounted for using empirical data, resulting in particle data that's aligned to our hazard threshold particles. For example, mantitol nets often miss fibers, as they can pass through the nets lengthwise. We corrected for this systematic bias with grab sample fiber data. To explain the further corrections, I first need to explain the laboratory process for analyzing microplastics. A sample taken in the field is extracted, and the particles are identified and categorized using microscopy. Pictures and measurements can often be taken, and particles are then examined for their chemical composition using light-based spectroscopic techniques or similar. In the case of the San Francisco Bay study, we used Fourier transform infrared spectroscopy to characterize the chemical composition. Unfortunately, this process can take a long time if there are many particles, especially if the particles are assessed manually, which is how we did it for this study. We used spectroscopic subsampling to estimate percentages of particles that are indeed plastic. We accounted for the fraction of particles in our assessment. The third and most important correction we had to make dealt with the size of the particles. Mantitol nets capture particles about 330 microns and larger. And because microplastics in the environment occur in exponentially higher abundances with smaller sizes, this sampling technique misses about 99% of the particles between 1 and 5,000 micrometers. All of the corrections we performed were done using the best available methods, including the use of empirical data, derivation of probability distributions, and Monte Carlo modeling. This resulted in a total correction factor of 2,819, with a lower 95% confidence interval of 336 and an upper 95th percentile of 17,966. After correcting and rescaling the data to match the effect thresholds, we can then compare to our management framework. We can see here that the majority of samples taken within the central part of the San Francisco Bay exceed our most conservative threshold, which would recommend additional investigative monitoring, with some samples exceeding these higher tier thresholds, including management planning and source control measures. Outside of the Bay, you can see that there are samples that are below even the most conservative threshold, suggesting that there's not likely to be risk in that area. The probabilistic assessment revealed relatively high uncertainties due to the corrections, with as few as 25% and as many as 100% of samples exceeding the most conservative risk threshold. Corrections for lost fibers and mantitol nets introduced the highest uncertainties into this assessment, with spectroscopic subsampling introducing the least amount of uncertainty. Based on these findings, we recommend that future monitoring studies collect samples using inline filtration pumps with high volumes and small filter sieves to avoid necessary corrections for fibers and reduce uncertainties due to size rescalings. Now I'll shift gears and talk about microplastics in drinking water and the human health risk assessment conducted by our Health Effects Workshop. To assess risks to humans through drinking water, we performed a literature search and found 29 in vivo mammalian studies that had exposed animals to microplastics orally. After screening for quality criteria pertaining to experimental design, particle characterization, and an applicability for risk assessment, we identified 12 studies that we deemed fit for purpose for risk assessment. We then subjected these 12 studies to additional scrutiny through expert review. While all of the quality screening criteria are important, we identified a subset of criteria that are critically important for additional review. Approximately 40% of studies could not be used for dose response assessment due to their use of fewer than 3 exposure concentrations. Of the studies deemed reliable and relevant for risk assessment, 4 reported effects on male reproduction, 2 on female reproduction, and 1 on the liver. Studies consistently showed adverse impacts to sperm deformity and count, as well as decreased testosterone in male rodents, all of these occurring in a dose-dependent manner. In female rodents, 2 studies reported lower anti-Mullerian hormone concentrations, which is a reliable biomarker for impaired reproductive success in mammals. For microplastics as a class, the linkages between the mechanisms and the observed effects in rodents are poorly understood. This makes extrapolation of effects observed in a single particle type, for example polystyrene spheres, to a diversity of microplastics highly uncertain. Adverse outcome pathways are lacking for microplastics, but some commonly observed mechanisms include reactive oxygen species formation, oxidative stress, inflammation, cell death, lipid metabolism, and energy metabolism. To estimate concentrations at which impacts may occur in humans, we analyze dose-response data from the reliable endpoints using EPA's benchmark dose modeling software. Point of departures for each critical endpoint are shown here, with different color points corresponding to different studies. The most sensitive effects were for testes weight and anti-Mullerian hormone concentration, which again is a biomarker related to female reproductive impacts in mammals. We also observed a similar range of concentrations in which other effects occur, especially on male reproductive impacts such as sperm viability and maturity. We know that humans are exposed to microplastics, as we've recently characterized them in the lung, blood, placenta, and breast milk, and found them in the majority of people tested, but we need more quantitative estimates for exposure to characterize risk. Quantifying exposure is a key component to deriving a health protective concentration in drinking water. A recent meta-analysis estimated exposure to humans through various fruits and found a high variability in uncertainties, ranging approximately 3 orders of magnitude within the interquartile range. This assessment estimates that most humans ingest about a grain of salt of microplastics per week, with less than 1% of the population ingesting the equivalent of a credit card's worth of plastic per week. This assessment is likely an underestimation, however, as less than 20% of dietary sources have been characterized thus far. The high variability and uncertainty in this exposure characterization presents challenges for deriving a quantitative risk threshold in drinking water, as estimating the relative source contribution from drinking water is an important driving factor. Despite our findings of consistent impacts to male reproduction in rodents, significant data gaps currently prevent a reliable risk assessment of microplastics to humans through drinking water. We identified 3 classes of problems, including an inadequate effects database, with most studies poorly characterizing particles and using limited polymers, shapes, and sizes that are marginally representative of the complex exposures that humans are actually exposed to in the environment. Additionally, while there is evidence for oxidative stress and inflammation being linked to the apical endpoints we saw in mammals, uncertainties remain. Understandings of mechanisms of toxicity are necessary for extrapolating effects across diverse particle types within the microplastics contaminant suite. Lastly, an incomplete exposure database represents an important data gap, with limited information for microplastics in our diets and no harmonized drinking water data. The State Water Board is monitoring and supporting additional science needed to assess the human health risks of microplastics. In particular, we need exposure data on what we are drinking, breathing, and eating, and we need high-quality laboratory toxicity studies to determine how the effects are occurring and at what concentrations they occur. In the United States, we are aware of only one project funded by the federal government to address these knowledge gaps, totaling over $100,000. In contrast, the European Commission has awarded over 30 million euros to research this issue. We believe that additional research funding is urgently needed in the United States and elsewhere to determine exposure and the risks of microplastics to humans. Now that we've covered the health research, I'll talk about the state's efforts to develop standardized methods and implement an accreditation program for laboratories. When Senate Bill 1422 was passed, no harmonized methods for monitoring microplastics were available, so the State Water Board developed our own. Through a partnership with the Southern California Coastal Water Research Project, we developed standardized methods using 2 types of instruments applicable to drinking water, ocean water, fish tissue, and sediment. Method performance was evaluated through an inter-laboratory validation study involving 26 laboratories from 7 countries. To assess method performance, laboratories were sent blind samples that contained realistic mixtures of particles found in drinking water. 4 common plastic polymers were used, each in varying amounts in 4 size fractions in 4 different shapes, ranging in size from 1 to 1,000 microns. Additionally, false positive materials like sand and shell fragments were added to test the robustness of the laboratory's identification methods. As proficiency testing samples for microplastics were not commercially available at the time, we worked with an organization in Norway to develop these samples specifically for this project. First, let's look at the overall findings from this study. On the y-axis, which is in log scale, is the percentage recovery, with a dotted line showing the 100% mark. The closer the recovery is to 100%, the more accurate the measurement. The grouping of data reflects the precision. Looking at the pane on the left for total particle recovery, there was relatively low recovery and low precision. But this is not the whole answer. Let's look at the second panel from the left, which breaks the data up by size fraction. Here we can see that both precision and accuracy are good for particles larger than 20 microns. And in the third panel, we can see that higher recovery was expected was found for blue and clear particles, and lower than expected recovery was found for orange and white particles. A similar trend is seen with shapes on the final panel, with higher than expected recovery for fibers and lower than expected recovery for fragments. Based on the overall strengths and limitations of the assessed methods, we feel that we have 2 methods that are ready for use for drinking water analysis, including infrared and Raman spectroscopy. IR and Raman spectroscopy are highly accurate for identifying plastics, while optical microscopy has limited overall accuracy. The average measurement time per sample is higher for Raman than infrared, and infrared instruments are generally cheaper than Raman. The consumables are also more expensive for Raman on average. The main strength of the Raman is that it can identify smaller particles with an approximate lower size limit of 2 microns. In addition to having a standardized method, laboratory accreditation is key to obtaining reliable monitoring data. California's Environmental Laboratory Accreditation Program will be providing accreditation to qualified laboratories starting this fall. Proficiency testing samples are invaluable tools to assess laboratory performance. We're partnering with the National Institute of Standards and Technology to develop a range of PT samples to meet our data quality needs. Now that we've defined microplastics, developed a standardized analytical method, considered the health effects for humans, and can accredit qualified laboratories, it's time to go out and do some monitoring. In designing a sampling program to assess exposure to humans through drinking water, it's important to know how these particles move through drinking water treatment plants. Treatment removes microplastics based on the filter sizes. Most drinking water treatment plants that have surface water for their sources use filtration methods to remove particles already, and may incidentally be removing microplastics. Flocculation and sedimentation can remove particles as small as 1 micron, about the size of a red blood cell, and more advanced filtration methods such as microfiltration and reverse osmosis can remove even smaller particles. As I showed earlier from our method validation study, we can reliably quantify microplastics as small as 20 microns. So we can't currently monitor for microplastics that are small enough to make it through most treatment techniques. We are working to improve these methods, and in the meantime, our current methods can be used to characterize the contamination in the source waters, which will be an instrumental first step in determining contamination and potential exposure to humans through drinking water. Microplastics, like other particles, have predictable behaviors in the environment. As microplastics get smaller, they become exponentially more abundant, and this relationship is illustrated well through this histogram, which may be likened to an iceberg. If one knows the shape of an iceberg, they can measure the visual portion and reliably estimate the portion below the surface. For microplastics, we have decent understandings of size distributions in freshwater down to 1 micron in size. We are taking an iterative approach to monitor for microplastics in drinking water. The first year is a pilot research phase focused on closing scientific gaps and building monitor infrastructure. Following the pilot phase, we will issue monitoring orders to a limited number of systems to document contamination in source waters. As part of this phase, we hope to develop a rapid surrogate monitoring method that can indicate potential contamination at cheaper costs. The final phase will build off of this second phase and explore additional sources and, if improved analytical techniques are available by then, monitor in treated drinking water. Currently, the only standardized sampling method for microplastics is ASTM D8332220. This method involves connecting a flow meter and flow control valve to a sampling tap, then filtering water through a stack of sieves. As the sieves are open to the air, they can become contaminated by atmospheric deposition and fibers from the operator's clothing, therefore requiring a field blank and potentially the subtraction of background contamination from results. During the pilot phase, we will compare this ASTM method to a newer method that utilizes inline filtration, therefore eliminating the potential for background contamination during sampling and eliminating the need for a sample blank and background subtraction. Data collected by the state's investigative monitoring could yield important information for the sources of contamination and the relevance to human health risks. The complexity and diversity of microplastics characteristics yields large and rich datasets. However, no harmonized data reporting standards currently exist, which could provide challenges for utilizing and comparing the data that we receive. In partnership with the Moore Institute for Plastic Pollution Research, the State Water Board is developing an open source data intake platform for microplastics that would harmonize the data reporting and integrate with existing data repositories that the state has and elsewhere. This is a draft schematic of this data processing pipeline. The project will be completed within a year, and because the code will be open source, anyone from around the world will be able to use and adapt it for their own needs. Now that I've discussed what we're doing with microplastics in drinking water, I'll circle back to our larger management strategy to address microplastics in the environment. To assess and manage risks of microplastics in the environment, the Ocean Protection Council and the State Water Board adopted a strategy earlier this year that takes a two-pronged approach, which includes multi-benefit, no-regret solutions to act on the problem now, and science to inform future action. This strategy is bold and ambitious and will require input from many stakeholders across all sectors. The strategy's science needs calls for monitoring of the environment, assessing risks to prioritize additional monitoring and interventions, characterizing sources and pathways into the environment, and then developing and implementing intervention techniques to mitigate impacts. This is an iterative process that will refine itself over time. An example of how this strategy is playing out is from monitoring data obtained from a recent study in San Francisco Bay. Monitoring in stormwater revealed high loads of rubbery fragments. These rubbery fragments were determined to be tire wear particles, which are known to shed at high rates during the normal wear and tear of driving. Luckily, solutions already exist to capture particles and other contaminants in stormwater. For instance, rain gardens and green stormwater infrastructure, like what's shown here, are engineered systems that are often used in urban areas. One type of green stormwater infrastructure, a bioretention cell, was found to capture 84% of microplastics that run off from urban stormwater. The state will implement this intervention strategy where appropriate and monitor to determine its relative success. While many think of plastic pollution as a water or just an ocean issue, we are discovering that a significant portion of the contamination is actually happening in terrestrial environments. For instance, microfiber emissions to land now rival those to land and water and are rising quickly. The majority of this contamination is from polyester fibers from our clothing. A recent study estimated that 2.2 kilotons of synthetic microfibers were generated by apparel washing in California in 2019 alone. Of that, 74% was estimated to enter terrestrial environments through the application of biosolids to farms. Plants can uptake and accumulate small microplastics through their roots, as visualized by these green fluorescently labeled microplastics. When plants are exposed to microplastics in soil, they grow more slowly and may produce less fruit. The impacts of terrestrial environment contamination may not be occurring equitably, as in California, the contamination is mostly occurring in low-income communities in our Central Valley, which receives the bulk of contaminated biosolids that are generated by high-income communities and urban centers. In addition to potential impacts to food security, microplastics are known to impact dozens of terrestrial species. A recent global risk characterization estimated that at least 5% of terrestrial species are at risk in 63% of soil samples monitored worldwide. Solutions already exist to reduce microfiber loadings into wastewater treatment plants and therefore into biosolids and land application. Installing washing machine filters in 10% of homes in a small town in Canada resulted in a significant reduction of microfibers in the wastewater treatment plant effluent. Based on this and other findings, France is currently requiring that all new washing machines come with microfiber filters, and the California legislature is considering a bill that would require similar regulations. If you are conducting research on microplastics and would like to learn more about California's efforts, I invite you to join our working group. I lead the Microplastics Monitoring Subcommittee of the California Water Quality Monitoring Council, which is a local and global community exchange of information and data for microplastics monitoring methods and tools. We meet quarterly and everyone is welcome to join. Plastic pollution research advances rapidly these days, and keeping track of important findings is dizzying to say the least. That's why I developed the plastiverse.org, which is a free website that aims to remove barriers on plastic research. The Plastiverse is a hub for assets and information exchange on plastic pollution research, and contains an explorable toolkit with data, tools, and more to help researchers and policymakers make evidence-based decisions. Thank you all so much for your attention. I look forward to connecting with you here in person and over Twitter.